Hello, my name is Mr. Kennedy, and for this Forth Valley West Lothian RIC supported study resource, we are going to go through National 4 Biology problem solving techniques, specifically looking at types of problem solving questions that you can expect to find in the National 4 UASPs that you will sit in class. These techniques will also prepare you for Level 5 or National 5 study in the future as well, so it's a good foundation to start a solid base from. So, our learning intention for this lesson is to develop our understanding of how to answer problem solving questions. Our success criteria are firstly is to be able to calculate cell division rates. Secondly, to develop skills for identifying information from graphs, to identify limiting factors of different cellular processes, and finally, to calculate simple whole number ratios. There are certain lesson symbols that you'll see throughout the lesson. So throughout the lesson, you will see various symbols on the slides to indicate what you should do. Here are symbols which may be used and what they mean. So there is listen, speak and repeat, read, write and copy, correct your work, and pause the video to complete the task. Of all of these options, I'll quickly, if you bear with me 2 secs, I'm just going to get my pointer to work. Of all the options that we'll use this read function, write and copy, and pause and correct your work. These will be the most frequently used throughout this presentation. And where you'll find them is in this right-hand corner here, bottom right-hand. Things that you'll need for this lesson. So you will need either something to write on, be it a jotter or some paper. You'll need something to write on that jotter or paper with, so a pencil or a pen. And you'll also need a calculator. So you may want to use your calculator that you use in school, or if you've not got that to hand or you don't seem to know where it is, then use your calculator on your phone, okay? So, in this lesson, we're firstly going to look at cell division. So why do we need to make new cells? Well, when we're still young, our body needs to produce new cells to help us to grow. Every time a cell dies or is damaged, a new cell is needed to replace it. Take a note of this slide just now. Now, some examples of cell division happening are we are not born the same size that we end up throughout the later stages of our life. So we grow constantly throughout our lives until we reach adulthood. So we are undergoing cell division, which makes us taller and makes us wider as well. If you have ever fallen and skinned your knee and you've got a scab on your knee, you'll notice that the scab doesn't stay there forever. So your body is going through cell division, and because those cells have been damaged and they've died, the body is then going through mitosis or cell division to be able to get rid of those dead cells and replace them with new ones. You may want to take a note of one of those examples just to make it a bit easier to remember in your head. So most animals and plants start off life as just a single cell. We all started as 2 cells joined together to form a single cell. Sperm and egg joined together to form a single cell. But these cells grow to become adults containing billions and billions of cells. So we go from one cell through to billions of cells. So we go from being unicellular with our one cell at the start to multicellular, being made up of different cells and different body systems. The type of cell division that makes animals and plants grow is called mitosis. So the cell division that we're going to look at is called mitosis. In mitosis, a parent cell divides into 2 identical daughter cells. These daughter cells divide into and so on. So if you look at the picture in the bottom right just now, you can see that we've got our parent cell. This is like the original cell that is the starter. when the cell splits, it goes through mitosis, and this here is the exact same as this. So these two are called the daughter cells. So the parent starts off, it splits into two identical copies to form two new daughter cells. Now, the daughter cells are exactly the same as the parent cell. Okay, so all these cells here are the same. So we go from one cell and it splits into two. So if we have a situation where a cell divides every 30 minutes, how many cells will there be after 1 hour? Well, basically, we're going to have 4 cells after 1 hour. Now, please do take a note of this question and the actual diagram so that you have a detailed note of how this process works. So we know that the cells divide every 30 minutes. That's the information they've given us, and they want us to figure out how many are going to be there after 1 hour. So the best thing to do is to start at 0. We know that we have one cell to start with. We start with a cell, we're quite happy, a cell being 1. So our first cell at 0 minutes hasn't divided yet, but we know after every 30 minutes it's going to divide. So after 30 minutes, it's divided into 2 new cells. Then we know we've got to divide it again to get to 1 hour. So we go from 30 minutes, add on 30 minutes to 1 hour, and these 2 cells have done the same thing this one's done. This one's split into 2, and this one's split into 2. So the cells that we have after 1 hour, we've got 4. Okay, so we started at 1 at 0 minutes, 2 after 30 minutes, and 4 after 1 hour. So for example, we have got our first one here at 0 minutes, and we're going to try and figure out this example. So every 20 minutes it divides, and we need to see how many after 100 minutes. So at 0, we've got 1. 20 minutes later, we've got 2 divided. After 40 minutes, we've got 4 cells. 1 hour, we've got 8 cells. 1 hour 20, we've got 16. And then 1 hour 40, which is just an hour plus 40 minutes, so 100 minutes, gives us 32. So our final answer for this question is 32. We've got 32 cells after 100 minutes. Okay. So what I'd like you to do is attempt the following question. So a cell divides every 5 minutes. How many cells will be present after 1 hour? Is it A, 4096, 2048 for B, C, 12 cells, or D, 512? I would like you to take 3 minutes, pause the video, take 3 minutes, read through the question again, get all the information, and then use that to answer the question. Pause your video now. Too complicated. It makes more sense to even just look at the question first. So, a cell divides every 5 minutes. How many cells will be present after 1 hour? Well, 5 minutes is our time it's going to take. So we know every 5 minutes we need to get a cell dividing. We need to do that for 1 whole hour, so we need to do that. So let's start the process. So at 0 minutes, we know we've got 1 cell. After 5 minutes, that's our first division, so we've got 2 cells after then. 2 splits after 10 minutes into 4, and then another 5 minutes later, we have 8 cells. This carries on and keeps going. Now, a trick for doing this is if you have your calculator with you, whatever number you had before, times it by 2. So if it's 1, times it by 2, that gives you 2. 2 times 2 gives you 4. 4 times 2 gives you 8. 8 times 2 gives you 16, etc, etc. After 40 minutes, we're down at larger numbers. We're at 256 currently at 40 minutes. 256 times 2 gives you 512 after 45 minutes. Times that by 2, 50 minutes, we've got 1024 cells. 55 minutes, we times again our answer before, that gives us 2048. And then finally, after 1 hour, we have got 4,096. So to conclude, we know that A is the correct answer because we sat and worked it out. So we can be confident in our answer, but we definitely know that A is the correct answer in this situation. So here's another one for you to try. So a cell undergoes mitosis every 5 minutes. Calculate how many cells will be present after 40 minutes. If you use the information from the previous question, this should not take you too long to do. So I will give you 1.5 minutes this time. What I'd like you to do is pause the video for 1.5 minutes and then carry on to the next slide after that. So use the information from the previous question, read through the question, and then once you've done that, have a try at going over it in the next slide. So a little bit different compared to the last slide with the way it's laid out, but it's the same principle. Every 5 minutes we've got our division happening, and we know that our final time that we need to get to is 40 minutes. We know at 0 minutes we've got 1 cell, and it splits every 5 minutes. So if we count it up, times it by 2 every time that we go along, we know that we've got 256 in this situation. So the exact same thing is happening as on the last slide, but we're just not going all the way up to 1 hour. We're only going to 40 minutes. Okay, here's another one for you. A cell divides every 20 minutes, so we've changed the question slightly. How many cells would be produced from one original cell at the end of 2 hours? So 20 minutes is every time it divides, and we need to go all the way to 2 hours. So we need to figure out how many minutes are in 2 hours to be able to answer this question. So I will give you 3 minutes this time, double the amount of time as the last question to answer. Pause the video, 3 minutes, allow yourself, and then carry on to the next slide. Okay, so as I said in the previous slide, we talked about the time trying to put our 2 hours into minutes. So 1 hour equals 60 minutes. So therefore 2 hours is 60 times 2 and gives us 120 minutes. So we know that if we have 120 minutes, we need to find out how many times 20 goes into 120. We know that 20 times 6 gives us our 120. So it's 6 times that we need to find this division. So if we start at 0 again with our 1 cell, after 20 minutes it's divided into 2. After 40 minutes it's divided into 4. After 1 hour it's divided into 8. After 80 minutes it's divided into 16. 100 minutes it's divided into 32. And then finally, after 2 hours or 120 minutes, we've got 64 cells in total. Okay, this one is a lot trickier. So we've changed the starting value, and we're not wanting to know how many cells are at the end, we want to know how long it takes. So starting with 1,000 cells, so instead of starting with just 1 cell, we're starting with 1,000. These cells, these 1,000 cells, divide every 15 minutes. How long until there are 15,000 cells present? So I'd like you to take, I'm going to give you 5 minutes to have a bash at this one, because this is a lot harder than the previous question. So we start with 1,000 instead of 1. Every 15 minutes, we know they divide, and we want to find out how long until there are 15,000 cells. So take your 5 minutes now. Okay, so we can see our starting value, 0 minutes, we have got 1,000 cells. So just as the question has told us, nice and easy, we've got that information put in. Every 15 minutes it divides, so 1,000 cells divides into 2,000 cells, just like how 1 divided into 2. We're just adding on the zeros at the end. 15 minutes plus 15 gives you 30 minutes. we've got 4,000 cells after 30 minutes. Another 15 minutes goes along, we've got 8,000 cells because this is doubled again. And then finally, this one here, 60 minutes, we can see that we've got 16,000 cells. So we know that 15,000 cells, 16,000 is greater than 15,000. So this is the number where we know that there are 15,000 cells there. So 60 minutes, 1 hour, is our final answer for how long it took for us to get these 15,000 cells present. And this is the final question that we're going to do for cell division before we move on to the next part of this lesson. So, 200 cells are undergoing mitosis. The cells divide every 30 minutes. How many cells will be present after 3 hours? So remember your steps. Look at your starting value, 200 cells. Look at how many times or how often it starts to divide. Now it divides every 30 minutes. And how many cells will be present after 3 hours? So how many times does 30 minutes go into 3 hours? Once you've got that information, then you can figure out the question and work it out with your numbers. Nice and easy question when you break it down. So this time, 3 minutes, not as generous as last time, but I don't think you need as much time because it's not as hard. So take your 3 minutes now and go over it in the next slide. Okay, so taking the information that the question has given me, I have said that at 0 minutes there are 200 cells. So 0 minutes, 200 cells, because that's what the question tells me. The question's also told me that every 30 minutes the cells divide. So after 30 minutes, these 200 become 400. We need to find out up until 3 hours. Now we know that 30 minutes is half an hour. So 2 halves make a whole. So 3 times 2, because there's 2 halves in 1 hour, gives us 6. So we're going to have 6 times that it's going to divide. So we go from 400 at 30 minutes to 800 at 60 minutes because it's doubled. Then we add on another half hour and it's divided again into 1,600. Again, it's divided 3,200 after 2 hours. 2.5 hours, we've got 6,400. And then finally, after 3 hours, we have got 12,800, sorry. So our final answer is 12,800 cells for this question. Okay, so we're going to move on just now. So we've looked at our answering cell division questions. We're going to look at something slightly different. We're going to interpret information from graphs. Now this is something that you will need to be able to do for your UASPs, and it's quite a difficult skill at first until you get used to the method of doing it. So graphs can appear confusing at first, and the best thing to do is look at the scales at the side to figure out what they are measuring. So if we look at the scale along the bottom, we can see that it's measuring temperature, and we can see that it's going up from 0 to 10 to 20 to 30 to 40. So we know that every one of these lines here is 10. It's nice equal spacing and it's going up by 10 every time. So they've given us this nice clear information along the bottom. If we look to this axis here, we've got rate of clotting units per minute. Now we don't need to worry about what that means. That's not important information, but we know that that's what it's measuring. And if we look, it goes 0, 10, 20, 30, 40, etc. until it gets all the way up to 90 at the very top. And again, it's nice equal spacing. So we've got one measurement along here and another measurement along here. On this graph, you've probably noticed that this is a bit weird looking compared to what you would normally see. So we've got 2 lines which are different as well. So this line here represents young camels, and then this line here represents young cows. So we've got 2 things getting measured for the same factors, temperature and rate of clotting, in the one graph. So we've got two animals in one graph for the same thing, okay? So don't get confused because they are just getting measured for the same thing, but this line here is for the cow and this line here is for the camel. So from doing this, it appears that the graph's measuring the effect of temperature on the rate of clotting for different types of milk. So that's what we can assume from this question so far. So I'd like you to have a try at the question just now. At which temperature is the rate of clotting using young cow rennet 70 units per minute? So you need to look at the graph, look at which line is the young cow, and then find 70 in the graph and tell me what temperature it goes down to. I'd like you to take 3 minutes to have a go at this. Use the information from the question and the graph to help answer this question. 3 minutes, pause your screen now. Okay, so the first mistake you may have made is that you may have used this line here. Now, it is important to read all the information on a graph. So this line here was to do it with the young camel, and this question asked about young cows. So this line here, we do not need to bother with at all. The solid black line, get rid of it. It's not important. The one that is important is this line here. Okay, so this line here is the important one, and we need to look at this one. Now, it tells us that we're looking for the temperature, which is the rate of clotting using young cow rennet 70 units per minute. So we can see that rate of clotting units per minute, and we go along to 70. Now, where it meets the line is here, where my red box is, and we know that that's 70. We're going to take this line down, and as we go down, it's nice and easy, takes us all the way down, and voila, we can see that the temperature is 20 degrees Celsius. So we followed it down, nice and easy, using the information in the question, and we've found that the temperature, the rate of the clotting is 20 degrees Celsius. So if you managed to get that well done, if not, I hope this makes it a little bit easier when you're looking over these types of questions. So we know that the final answer is 20 degrees Celsius. Okay, so using that practice that we've just done, I would like you to have a go at this question here. So this graph is looking at the distance between a lamp and Elodea. So you might have seen an Elodea bubbler in class where you've got a lamp and you've got an underwater plant and you're counting the bubbles. So this bottom line talks about the distance between the lamp and the Elodea in millimeters. And this section here, going up this axis, looks at the rate of photosynthesis and it's counting the number of bubbles per minute that the plant is giving off. So the question asks, what was the rate of bubbling when the lamp was at a distance of 200 millimeters from the Elodea? I'd like you to take 3 minutes again, the same amount of time as the last question. I've given you less information and guidance than the last time, so 3 minutes should be fine for you to have a bash at this. On you go. Okay, so let's go through this answer here. So we know that the information that they're given us in the question is that it starts, or we're looking for a distance of 200 millimeters. So we're looking for the one that's got millimeters along the bottom, which is this bottom line here. And let's look for 200. Now 200 is at this point here. So we're going to follow it up until it reaches this line here. And we can see, okay, we've met our line. Now we need to go along to our x-axis. And as we follow it along, we can see that it meets at 15. So at 200 millimeters, it is causing 15 bubbles per minute. So our final answer is 15 bubbles per minute. So well done if you managed to get that. It was a trickier question for this one. Okay, so when we're looking at these kind of graphs, there are important things to think about. So you can see that there's three graphs there, and we're going to talk just now about limiting factors. Now, a limiting factor is something that limits the rate of a reaction, and when we're talking about different reactions, we can talk about different limiting factors. For photosynthesis, there are three main limiting factors. There is light, so the light intensity, how intense the light is for the plant. There is carbon dioxide concentration, how much carbon dioxide the plant has to breathe in. And then finally we've got temperature. Now, you may have looked at enzymes before, and if you may remember that enzymes need temperature for them to work at their optimum or their best. So we can see these two graphs look the same. They kind of increase and then level off. This one increases and levels off. So its limiting factor is at this point there's low light, so it's limiting it. As the light increases, this is like a high point where it's got the most light, but we can see it levels off. So that basically means that it is not getting any more light. It has got as much light as it needs, and something else is stopping the rate of photosynthesis. So our limiting factor here could either be carbon dioxide or temperature, but at this low light down here, it's going to be light, obviously. Same thing with carbon dioxide. Low level of carbon dioxide, we've got a low rate of photosynthesis. As the carbon dioxide concentration increases, so does our rate of photosynthesis. But there's only so much it can take in, and then it levels off again. So when it levels off, it could be temperature, or it could be light that is limiting this process. Temperature looks different. It's not got a leveling off phase in it. So when we see it, it goes all the way up until we talked about this word optimum. Now the optimum is like the high point in the graph, the temperature that it works best at. Now if it was in humans, then our optimum temperature is body temperature, which is 37 degrees Celsius. So we would be going up 37 degrees Celsius. Any hotter than that would cause it to drop off. Now, with light and carbon dioxide, you can see that the graphs level off. This because the organism, the plant, can't take in any more of that substance. The temperature graph reaches its optimum up here, where it's working at its best. If it gets hotter than this, then we can see the activity goes down dramatically. It drops down straight away. This is because the enzymes needed to complete the reaction are known as denatured. Their shape has changed and they're no longer able to be useful for that reaction. So, I'd like you to take a note of the following slide. Now, we talked about photosynthesis and its three limiting factors. We talked about how the temperature needs to be warm. There needs to be enough light for the plant to be able to undergo photosynthesis. And it also needs this carbon dioxide concentration. So plants breathe in carbon dioxide and they need it to help them go through photosynthesis. Respiration is a little bit different. Respiration doesn't need light because it can happen in humans, and humans don't need light to make food. They don't make their own food. So we need a nice warm temperature so that our enzymes are working comfy at their optimum. But instead of carbon dioxide, we breathe in oxygen. So we're talking about oxygen concentration rather than carbon dioxide. Carbon dioxide for plants when they're going through photosynthesis, and oxygen for other organisms who are respiring. So, I've got a question here for you just now, thinking about these limiting factors. So what effect would increasing light intensity have on the rate of photosynthesis of plant growth? It's meant to say. It's not very well done. So, A, it would increase the rate of photosynthesis and decrease plant growth. B, it would decrease the rate of photosynthesis and decrease plant growth. C, it would increase the rate of photosynthesis and increase plant growth. Or D, decrease the rate of photosynthesis and increase the plant growth. I'd like you to pause this 2 minutes, read over the question. The answer is in the question. Okay, pause the video now. Okay, so if you had closely analysed that question, you would have gotten the information of increasing light. Now, if we look back to our graph, we know that as we increased the light, the rate of photosynthesis went up. So if we increase the rate of photosynthesis, then plant growth will increase. So as we can see from the graphs earlier, if we increase the amount of light a plant gets, then the rate of photosynthesis is going to increase, and this in turn allows the plant to grow quicker. The final part of this lesson is on ratios. Now, this is quite a difficult part of the National 4 paper and something that a lot of people struggle with. It's a concept that you'll probably have come across in maths, and it can be simple, but at times also complex to do with a lot of situations when ratio questions are used in biology. They are very wordy. So the best thing to use when doing ratio questions is to use a highlighter and highlight the numbers of different organisms. So in mathematics, a ratio indicates how many times one number contains another. So for example, if there are 8 oranges and 6 lemons in a bowl of fruit, then the ratio of oranges to lemons is 8 to 6. So we've got, for every 8 oranges, we've got 6. We know that we can simplify this by finding the smallest whole number which divides into both of these. We know that both of these numbers are even numbers. They are able to be divided by 2. It's basically what even numbers means. So if we divide 8 by 2, that gives us 4. And if we divide 6 by 2, that gives us 3. So a ratio of oranges to lemons would be 4 oranges to 3 lemons. So for every 4 oranges, there are 3 lemons. The following table shows the results from an Eladia bubbler experiment. Calculate the whole number ratio of the results from light intensity 3, which is this one here, with light intensity 4. So the bigger number first being number 4, and the smaller number second being number 3. Okay, so I'm going to give you 5 minutes to have a go at this question. Try to find your whole number ratio, and you're going to try to simplify it. So find one number that goes into both of these numbers. Your 5 minutes starts now. Pause the screen. Okay, so you've probably had your 5 minutes. So we're going to look at our question, and we're going to look at the results so we can find our whole number ratio. So we know that at light intensity 4, there are 25 bubbles per minute. At light intensity 3, there are 20 bubbles per minute. So our current ratio is 25 to 20. Both of these numbers are able to be divided by 5 because they're both in the 5 times table. So 25 divided by 5 equals 5. 5 times 5 is 25. 20 divided by 4 is 20 divided by 5, is 4. So our ratio is for every 5 there is 4. Okay, so our final answer is 5 to 4. We've simplified 25 divided that by 5, and it gives us 5. 20 divided by 5 gives us 4, and that is our final answer, 5 to 4. Okay, so using the same set of results, the following table shows the results from an Eladia bubbler experiment again. I'd like you to calculate the whole number ratio of the results from light intensity 1 and 2. So light intensity 1 being here, and light intensity 2 being here. So the same thing applies. You need to find a number that goes into both of those answers, and you need to make sure that you have got the bigger number at the start and the smaller number at the end. Okay. I will give you 3 minutes this time since we are getting used to doing this process. We've worked through 2 examples so far. So your time starts now, 3 minutes. Okay, so the first thing we needed to do was to figure out what numbers go with what units. So light intensity 1 and 2. Light intensity 2 has got 14 and light intensity 1 has got 7 bubbles per minute. So our current ratio is 14 to 7. Now we know that the times table that they both go into, 7 is an odd number and 14 is an even number. So we know they don't divide by 2. The next whole number that we can go to that they both go into is 7, because the 7 times table, 1 times 7 is 7 and 2 times 7 is 14. So they both divide by 7. So we can see that light intensity 1 is 7 and light intensity 2 is 14. Both of these numbers can be divided by 7. So 7 divided by 7 equals 1 and 14 divided by 7 equals 2. So therefore our whole number ratio is 2 to 1. For every 2 bubbles in number 2, there is 1 bubble in number 1. Nice and easy. That's our answer figured out for that question. Okay, so this takes us to the end of this lesson. So our learning intention at the start of this lesson was to develop an understanding of how to answer problem-solving questions. You should now be able to, using your success criteria, be able to calculate cell division rates, so looking at mitosis and how our cells divide. You should also be able to develop skills for identifying information from graphs, so looking at what is on your x-axis and your y-axis, and then being able to identify what information is on each and being able to make assumptions about what the graph is about. You should also be able to identify limiting factors of different cell processes. So things like your temperature, carbon dioxide concentration, and light intensity for your photosynthesis, and then oxygen and temperature for your respiration. Finally, you should be able to calculate your simple whole number ratios. So your simple whole number ratios are looking at comparing one thing to another. So it could be the number of oranges to apples. If you have got 14 oranges and you've got 7 apples, then you know that you've got, for every 2 oranges, you'll have 1 apple. So, some next steps for yourselves to have a go at. Try to attempt using these techniques through check tests on BBC Bitesize or the EduBuzz website. So I've put the QR codes for these 2 websites below. And these websites are quite useful because they contain all of the information that is in the course. But at the end of each section, they have got a check test, which allows you to basically check your knowledge to see if you've understood what you've just looked at in the presentation or whatever it is you've just looked at. So they're quite good to look at, and it might be worth revisiting this lesson again, then looking over your notes for each unit that you're currently doing in class, and then try not to use your notes to answer the questions. So revise first, then look at your notes, or revise and look at your notes, sorry, then put your notes away, and then use these websites and try to get some sort of understanding of how to answer them without having your notes. And this will allow you to be more confident when you go to sit your UASPs in class. Okay, so the QR codes are up on the screen now. You may want to use them to help with your further revision. Finally, I'd just like to say thank you and the best of luck for when you are doing your UASPs. I'm sure that you will do great, and if you use the techniques that have been in this lesson, then you will be great.